so that I can see all of you. Where's my... I'm setting up my screen, people, so give me a couple seconds to set up my screen. Okay. All right, I'm ready. You guys ready? Okay, so I think I talked about this picture already, right? You see this baby mouse, okay, baby mouse. This baby mouse received a gene from jellyfish, okay, from jellyfish. And this gene is called GFP. GFP, that's the gene. It's called the green fluorescent protein, okay, GFP. Now, they get it from the jellyfish because the jellyfish, they dive really deep in the ocean. And so when scientists discovered this glow in the dark jellyfish, they were wondering, how does it glow in the dark? You know, how does it glow deep in the water? So they took the jellyfish, removed the cells of the jellyfish, and then they looked for that gene. It took them a while. They found the gene, and they took the gene out. They splice it out. That means they cut it out, remove, and then they put it into a bacteria. They grew the gene, and then they insert it into the sex cells of the mother of the mouse. And then the mother mouse gave birth to the baby. And the baby carried that gene that glows in the dark. And then at the beginning, the baby did not glow. The baby was just like normal mouse. And then, but as it got to a certain age, the protein that's in the DNA of the mouse now, it started to produce the protein. This protein would create a fluorescent color, a bright, and it glows. Is it bright like this? Well, the answer is no. A lot of these pictures you see are Photoshop, okay? Photoshop, okay? So it's kind of like Instagram. You know what I'm talking about, guys? You guys know I'm talking about Instagram. When you look at those Instagram pictures of those people, influencers, they look like drop dead gorgeous. You know what I mean? The men and the women. But realistically, are they really that good looking in person? Take away the makeup, take away the eyelashes, the hair, the skin, the fake nose, whatever it is, the fake ears, and then they're just like everyone else. They're just like you and I, average, normal people, all right? So a lot of the pictures you see are Photoshop. Have I seen something like this? Yes, I have, but they were not this bright. But I shared with you last time that we do something like this in AP Bio, okay, AP class. So we take the jellyfish, this same exact gene that we buy at the store, but well, the scientific store. And then they send it to us, and then we take it, we put it into bacteria, and then we let it grow. We try to insert it in transformation. Remember that, transformation. Remember Griffith, right? Transformation. So the objective of this lesson is to talk about how your cells replicate the DNA, okay? Replicate DNA. So DNA replication copies the genetic information of a cell, okay? So how does it work? Okay, let's start from a little review, okay? Little review. Okay, now, all cells, okay, all the cells of your body, when you look at yourself in the mirror, they're all made of cells, but there are two different types of cells. There are sex cells and there are body cells, right? Sex cells and body cells. Both of these cells have to carry out cell division and before they do cell division, they have to make copies of their DNA. Now, most of your cells, it goes through a cell cycle. This is the cell cycle, okay? That's the cell cycle. Now, the cell cycle starts here. It starts with the interphase, this right here, interphase. Interphase makes up about 90% of the life of the cell. 90% of the time is in interphase. So you're looking at your skin, guys. You're looking at your skin. Most 90% of your skin is in interphase. It's preparing. It's preparing. So it goes through gap one, prepare, and then it goes to S, means synthesis, right? That's when the DNA doubles. So for humans, we have 46 chromosomes or 23 pairs, okay? So what happened is during the S stage, the 46 double. They double, okay? They double. They have to double because the cell is going to divide. And then once it's done, it goes into gap 2. Gap 2 is to check to make sure the cell is ready, right? Now, once that is ready, then it goes through division, mitosis or meiosis, and then bam, you start one cell, you have 2 cells. By the time you get to here, 2 cells. So most of these cells are in interphase. So this is gap 1 and gap 2. This is what the cell looks like, and it doubles up in size. 46 chromosomes, right here in the nucleus. By the time it's ready, gap 2, by the time it gets to gap 2, it still has 46. But instead of 92, which is double, right? Instead, the chromosome is like an X. Each of these are called sister chromatids. Now, when it's here, there's only one half. There's only half. But when it gets to here, it's 2 halves. Okay? It's connected. And it looks like this. Let me show you a picture. I know you're going to ask me. So here is your cell cycle. And what happens inside the cell, right? During the gap 1 and gap 2, the cell has to double everything. So if your cell has 4 mitochondria, it has to make 4 more. It has to be 8. Because when the cell divides, each of the daughter cell, has to have the same number of mitochondria. So if a cell does not double everything inside of itself, then if it divides, that means it has less, and then 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 the cell disappears? No, that's not possible. The cell has to maintain the size. And so this is what it looks like. You see, okay, you see this is a karyotype, okay? This is called a karyotype. It is a picture that shows you all the chromosomes inside, all the DNA inside a cell. Now, notice how this is chromosome number 1. Oh, let me make it darker. Chromosome number 1, number 2, number 3. You see, number 4, number 5, number 6. You see what I mean? There's 46 of them. You see what I mean? Now, there's only half, right? But during the S, it doubles. See, each of these, they make another copy of themselves. So this is the original, right? It makes another copy of itself. This is another half, identical. So they're attached at the centromere, right here. Attached at the centromere. So now, this is what the cell looks like after interphase, after S phase. So this is what a cell looks like here. After this, after the S, when it's here, it looks like this. It doubles. But the 2 pieces of chromosomal DNA are attached to each other. Two pieces of DNA attached at the centromere, okay? All right, so let's move on to how they divide, how they make copies. We're talking about the S, okay? That's the point. So during the S stage, synthesis stage of DNA during interphase, here is your DNA. Now, keep in mind that there are, how many chromosomes do we have inside our body cells? 46, right? I'm showing you only one. But all 46 do the exact same thing. Got it? There are 46 chromosomes. Each cell, body cell has 46. All of your body cells, you and I, we're all the same. We have 46 chromosomes. But I'm showing you only one chromosome instead of 46. But in reality, you have 46. Now, the process of replication, the process of DNA replication is completed by proteins, enzymes inside your cells. Now, these enzymes it comes from the food that you and I eat. The food that you and I eat, they are assembled from the food that you and I eat. Now, the DNA produces the enzyme. And the enzyme are assembled by the raw material that are found from the food that you and I consume. So, the DNA, what happened is, at the start of the process, what happened is it has to uncoil. It has to open up. Okay, it has to open up. So notice how the double helix, it opens up. It opens up here. See, we could say it unzips. Okay, it unzips. Now, this is all done by the work of what we call enzymes. Okay, The enzyme is called helicase. The helicase is the enzyme that unzips the DNA. Unzips. Open it up. So what it is, it cuts the hydrogen bonds. It cuts the hydrogen bonds. It cuts the hydrogen bonds so it opens it up. Now, once it opens up, then there's another protein that holds the DNA strand apart. So there's a DNA protein here, there's a protein here, and what happened is it holds onto the strand and it opens it up. Because if it lets go, it's gonna come back and it's gonna get spiral again. So that's another enzyme that does the work. Now, once it opens up, what happened is these nucleotides, this is the nucleotide for thymine, nucleotide for guanine, nucleotide for cytosine, nucleotide for adenine. So there are 4 different types of nucleotides, right? There's ATGC, A, T, G, and C. There's only 4 different types of nucleotides. Now, where do you get this? Where do you get this from? You get it from the food. You and I eat food. When you eat food that's healthy, it has a lot of this. It has abundance of these nucleotides. They are assembled by our body. So the food that you and I eat, it has all this stuff. So these food that you and I eat, it goes into the cell. And then when the cell is ready for replication, the enzymes, they open up the DNA. Once they open up the DNA, these nucleotides, they fly over. They attach to the DNA. They fly over. They attach. They fly over and they attach. They attach to it. Now, how do they attach? How do they attach to the double helix, right? So the way it works is, okay? The way it works is, it's like this. Let me give you a better picture, okay? Now, notice how the DNA opens up. Now, there are 2 strands, right? They open up because of enzymes. Helicase, DNA polymerase is another enzyme. Now, this is the main enzyme. This is like the brain of replication. And it's right here. DNA polymerase is right here. And right here, DNA polymerase. Now, what it does is it brings in the nucleotides. See these nucleotides? If this is a nucleotide is, let's say, a T, right? What happened is the A would fly in. Because A and T are complementary. If this next base is a G, then a C would come in. A C would fly in. You see what I mean? The base would fly in. And then if the next one is a C, right? That means the G would fly in. So what happened is your old DNA strand, the blue strand, that's your old strand. We call it template. Template means a copy. It's a copy to make other copies. So the orange, people, the orange is the new strand. The blue is the old strand. Your body uses the DNA strand, the old strand, as a template to make a new strand. Now, it goes in opposite direction. It goes in both directions. Let me clear this up, okay? So, replication occurs this way until it gets to the end. It also occurs this way until it gets to the end. It occurs in opposite directions until it gets to the end of the chromosome. Now, all 46 chromosome people, because you have 46, all 46 chromosomes do the exact same thing during the S stage of interphase, the S synthesis stage of interphase. With the help of enzyme, DNA polymerase is the brain. Okay, now eventually it gets to this. Eventually, your DNA is completely replicated. And so here is your DNA strand uncoiled. It opens up by the help of enzyme. Nucleotides then flies in. Nucleotides, they fly in and they attach. So if this is, let me change color. Oops. Let me use a green, okay? If this is a T, then the A would come in. You see what I mean? If this is a C, a G would come in, nucleotide. If this is an A T would come in. Because A and T are complementary, C and G are complementary. And so a C would come in. You see what I'm doing? See what I'm doing, guys? So this is the old strand, right? The old 2 strand. It opens up, both strands, and it moves in opposite direction. Replication occurs in this direction, and it occurs in the opposite direction as well, because you have a lot of DNA. You have billions of bases, and so therefore, replication has to occur in many, many locations in you and I, because we have so much DNA. Now, by the time it is done, it gets to the end. What happened is you have two strands. You have two strands, right? We call these sister chromatids. These are called sister chromatids. These are going to coil up, double helix, and they will be attached right in the middle. They're attached right in the middle here, called the centromere. That's why when you look at DNA, it's an X. It's an X. It's an X. It's an X because half of it is they're copies of each other. They're identical. This strand and this strand are identical to each other. Now, during mitosis, this is going to break, and so they separate into different cells, and that's how it works, okay? So now, what does it look like? What does it look like as it's completed, okay? So here I have a picture showing you what it looks like. Here is your original, the blue is your original. The orange is your new strand. So one old, one new, right? One old, one new. One old, one new. See? One old, one new. Now, this is what it looks like before interphase. This is what it looks like before S, okay? Now, this is what it looks like after S. You see what I mean? This is 2 chromosomes, right? This is also 2 chromosomes, but you notice how these 2 strands are double, right? Because they're identical to each other. They're attached right here at the centromere. The centromere, it's right here. Now, notice how one strand is old, one strand is new. One old, one new. We call this semi-conservative. Semi-conservative. It's the idea that in replication, inside each of your cells, half of the strand is old, the other half is new. Because half of the strand is the template, the copier, to make the other new copy. Half old, half new, okay? Half is the original strand, the other half is the new strand. Original strand, and new strand. And again, these are sister chromatids, okay? Sister chromatids, okay? And here's the new strand, which is the orange strand. Okay, guys, so far so good? Okay, now, all this is occurring during the S stage of interphase, okay? Now, so, how long does it take? That's the question, right? How long does it take? Well, in animal cells, in plant cells, in fungi, and protists, okay? Plants, animal, fungi, protists, right? Plants, animal, fungi, protists. We are called eukaryotic cells. Eukaryotic cells are you and I, plants, fungi, protists, okay? So eukaryotic cells are multicellular organisms. There's some that are single cell, but eukaryotic cells have certain special properties inside. Now, we tend to have more DNA than bacteria, right? More DNA. So for replication in eukaryotes, it is very fast, okay? It's very fast, and it tends to be very accurate because of the enzyme, DNA polymerase. So, see right here? DNA polymerase is creating replication here. So, this area is opening up. Notice how it's opening up. Replication is occurring this direction, and it's also occurring in this direction. It's moving this way, and it's moving that way. Now, in this picture, it's showing you only 3 DNA replication, but in reality, it could be thousands. There could be thousands of these sites of replication. Eventually, you have 2 strands of DNA. You got 1 strand of DNA, 2 strand DNA, and they're attached together right here in the center called the centromere. And we say this is a chromosome, but they are attached to each other, so we call them sister chromatids. So this is what I wrote. In eukaryotic cells, replication may begin at dozens or hundreds of places on the DNA molecule because we have so many. Then it proceeds in opposite direction until each chromosome is completely copied. Now, this is only 1 chromosome. How many chromosomes do you have? 46, right? 46 are carrying out replication at the same, guys, at the same time, at multiple locations, sometimes hundreds of these replication areas, okay? And then the enzyme DNA polymerase, the reason why you and I do not look like aliens, and we don't have like a third eye or, horns, things like that, we look weird, is because of this enzyme called DNA polymerase. It proofreads, it runs over the DNA one more time when it's done, it proofreads it to make sure that there's no error. Sometimes it might make a mistake, might make one mistake, but it doesn't happen often. So therefore, for humans, you don't see a lot of mutations. Unlike bacteria. Bacterias, they have a lot more mutations than we do. Number one, their cell cycle is a lot shorter than us, and so therefore they have a higher probability of a mutation than you and I. Versus you and I, I'll give you an example. Your skin, it takes, let's say you fell off your bike and you scratch yourself, right? You have a scar, right? How long does it take for that scab to completely go away? 30 days. It takes 30 days for your skin to completely shed. So you have 5 layers of skin, five layers of skin. For your 5 layers of skin to completely shed and that scab disappear, 30 days. When you're younger, it's like 20-something, 24 to 25 days, 26 days. But as you get older, it becomes 30-something days.